Okay, so in this video, we're going to be talking about oxidative phosphorylation. So under this topic, we're going to define what exactly is oxidative phosphorylation. So oxidative phosphorylation is a process in which ATP, so it's a process in which ATP is formed as a result of transfer of electrons. Okay, so transfer of electrons to oxygen by a series of electron carriers. So that's what we are going to dwell on today. We'll talk about ATP. How is ATP formed? What are the types of electron transfers? What are the types of electron carriers that are being referred to here? Alright, so let's just go into explaining what exactly is the mechanism. Alright, so the intermediates of the metabolic reactions like citric acid cycle, for instance, we had learnt that we had FADH as well as NADH. So these intermediates, they go into the electron transport chain, okay, and they donate electrons to the coenzymes NAD plus as well as FAD. Alright, now as they donate electrons, this is in order to form energy rich reduced coenzymes that are going to have NADH as well as FADH. Alright, so having said that, we are also going to see that in oxidative phosphorylation, as the electrons are being passed on down the electron transport chain, alright, they lose energy. As they are passing down the electron transport chain, energy is lost. So energy is lost, they lose much of their free energy, and part of this energy can actually be captured, okay? It's captured and stored by the production of ATP, okay? So that energy is lost in a form of an inorganic phosphate, so it's added on to ADP in order to make ATP. So that's how the energy is actually made. So the electron transport and the ATP synthesis by the oxidative phosphorylation, it proceeds continuously throughout all the cells, or rather all the tissues that have a mitochondria within them. So why is it that only cells that have a mitochondria are the ones that are able to undergo this process? It's because the components that are involved for the electron transport chain, they are found in the inner mitochondrial space. So there's what we call the inner mitochondrial space. Alright, that's what we find, and in this inner mitochondrial space we'll find several protein complexes that we call complex 1, complex 2, complex 3, complex 4, as well as complex 5. Okay, so each of the complexes are able to accept or rather donate electrons to relatively electron carriers. Okay, they all have the ability to donate or rather accept electron to electron carriers. So those electron carriers, some example are coenzyme Q. So we have coenzyme Q or ubiquinone, as well as cytochrome C. Okay, so these are the electron carriers that we're talking about. However, there is also something else that's formed as a result of the electron transport chain. So the electrons, in the end, they ultimately combine with oxygen. All right, so the combine with oxygen and protons, and the end product gives us H2O, which is water. So it accounts for the great proportion of the body's use for oxygen. So the body uses up oxygen even to produce water. So we do not only get water from what we eat, but even from the air that we breathe. There is a way in which it's converted to oxygen. So, however, there's an exception of complex 5. Of course, not regarding to the oxygen water production. So the exception is that complex 5 catalyzes the ATP synthesis. Okay, this is the one that catalyzes the ATP synthesis. Hence, it's referred to as ATP synthase. Okay, so this is something special about complex 5. All right. So we're going to talk about substrate shuttles that transport these reducing reagents like NADH, FADH, and so on and so forth. Okay, so the first one that we're going to talk about is the glycerophosphate shuttle. Okay, I think I'll even write it here. So this is the glycerophosphate shuttle. Okay, so this is one of the complexes, or not one of the complexes, sorry, it's just one of the shuttles that give us this, that we are looking for, the NADH that bears, it's an energy carrying molecule. So what exactly happens in this glycerophosphate shuttle? So what happened is that there are two types of transport shuttles that are employed in transporting these reducing agents, or rather electrons or protons. Okay, so one that that we're about to talk about is the glycerophosphate shuttle. The other one is called the malate aspartate shuttle, which we are going to talk about immediately we're done talking about the glycerophosphate shuttle. So under these shuttles, they are required since the inner mitochondrial membrane is impermeable to NADH. And remember, we've been saying every tissue that has a mitochondria undergoes oxidative phosphorylation. However, the inner mitochondrial membrane is actually impermeable to NADH. So the body employs a shuttle system. So to shuttle something is to make it pass through. Through means through which without it wouldn't have passed through, which is the glycerophosphate shuttle as well as the malate aspartate shuttle. So under the glycerophosphate shuttle, the NADH produced in the cytosol is used to reduce the dihydroxyacetone phosphate. This is the NADH that we're talking about. So it's used to reduce this molecule, and it reduces it to glycero 3-phosphate by the cytosolic enzyme, which is glycero 3-phosphate dehydrogenase. All right. Now this glycero 3-phosphate that we're able to see, this glycero 3-phosphate is the one that then enters the mitochondria. Let's take, for instance, this is our mitochondria, right? Okay, let's just undo this. All right. So if this is our outer membrane space, now we are in the mitochondria right here. Okay, so this glycerol 3-phosphate is the one that enters the mitochondria, and it's oxidized back to the dihydroxyacetone phosphate. So it's turned back to dihydroxyacetone phosphate. Remember, this is G3P. It's reoxidized back to the dihydroxyacetone phosphate. Okay, now the same enzyme that was used to reduce it is the one that is used to uh, oxidize, which is the glycerol 3 phosphate. Okay, and then because of this, we find that in the mitochondria, the FAD that's found in the mitochondria is during this reaction is rather oxidized to FADH. So this is the whole mechanism of the phosphate shuttle. The next shuttle that we're talking about is the malate aspartate shuttle. So like you can see the NADH in the cytosol, it reduces this oxaloacetate. So let me just use arrows. So it reduces the oxaloacetate. Like you can see, there is reduction, and reduction is an addition of a hydrogen atom. So NADH reduces oxaloacetate to malate. Okay, so it reduces it to malate by the enzyme that we call malate dehydrogenase. Okay, now this malate that has been formed due to the reduction process, it goes into the mitochondrial matrix. All right, so as it goes into the mitochondrial matrix, it's reoxidized back to oxaloacetate, and in the process, as it is reoxidized, it's the NAD is going to be oxidized back to NADH. All right, so the resulting oxaloacetate undergoes transamination. Transamination is an addition of an amine group. Okay, so it undergoes transamination in order to form aspartate, and what enables that is what we call aspartate aminotransferase. Maybe let me write it. So aspartate aminotransferase. So aspartate aminotransferase is the one that's causing this oxaloacetate to undergo transamination. Okay, so if oxaloacetate is converted to aspartate, it means aspartate can now go back to the cytosol, and the whole process will continue going on. All right, so this shuttle is a little bit different from the glycerol phosphate shuttle. So before we go further, let's just highlight that this is the malate aspartate shuttle. Okay, so this one is a little bit different because it's readily reversible. You can go back and forth, producing the same products. All right. Next up, we are going to look at what exactly happens. What are the procedures that occur in this oxidative phosphorylation? How are the how are the electrons transported, and so on and so forth? Okay, all right, so let's just find that page. Okay, so these are the complexes that we have mentioned earlier. We have complex 1, over here we have complex 2, complex 3, complex 4, all the way up to complex 5. So with the exception of ubiquinone, which is coenzyme Q, all the members of this chain are protein in nature. They're all proteins. All right. So the pair of electrons that are carried by NADH, along with 2 hydrogen atoms, are transferred to NAD dehydrogenase. Okay, in complex 1. So all this is embedded in the inner mitochondrial membrane. So complex 1 contains iron atoms that are paired with sulfur atoms. Iron is Fe, right? So this is what they are talking about. This is what is being talked about. It bears iron that's bonded to sulfur, as well as clusters of FMN. All right. So the electrons are transferred one at a time through this FMN. These are the two electrons. As oxidation reduction is happening, the two electrons are transferred via the FMN to the FES, or the iron sulfide. All right. So during the transfer of these electrons, there are four protons. Okay, four protons are translocated across the intermembrane space. I mean, across the inner membrane to the intermembrane space out there. Okay, and then what we notice is that these protons, the FADH, which is in complex 2, transfers electrons to coenzyme Q. It transfers electrons to coenzyme Q through this FES. Alright? And as it is doing this, there is an enzyme that we call succinate dehydrogenase. So, as all this is happening, succinate is actually converted to fumarate in the process. So, complex 1, there is a transfer of two electrons, as well as complex 2, there's a transfer of electrons from from FAD to coenzyme Q through this molecule, which is iron sulfide. Alright, now the amount of free energy that's released during the reactions is actually, it's not enough to pump electrons across the inner membrane. So the coenzyme Q, when it accepts the electrons, becomes it becomes reduced to ubiquinone, which is coenzyme Q H2. This coenzyme Q H2 is the one that's able to transport those electrons. So the coenzyme Q transfers electrons to complex 3. Okay, it transfers electrons to complex 3, and the remaining members of the electron transport chain are the cytochromes. All right, so the electrons are passed on along the chain, which is the coenzyme Q that we are talking about, to cytochrome C. So the rest of the electron chain carriers are cytochromes. Okay, so here we'll have cytochrome C being the one that's transporting to complex 4. So cytochrome C can only accept one electron at a time. Okay, so it's an exception in that way. In order to form cytochrome C in through complex 3, it has to do it twice, because cytochrome C is found on complex 3. So as it is receiving electrons from complex 2, it can only do that. It receives one at a time, so it has to do it twice. Alright, and then the transfer of these electrons through the complex 3 and complex 4, it results into pumping of four protons and 2 protons respectively into the inner mitochondrial space. So this cytochrome C is associated with the 4 of the inner membrane, like the coenzyme Q that we had talked about at this stage. Alright, so at this point we'll go further to talk about so at this point, cytochrome C oxidase. So cytochrome C oxidase, which is complex 4. This is complex 4. It's the only electron carrier in which the heme ion can react directly with oxygen. Okay. This is the only one where the heme ion can react with oxygen. So this is exactly what's happening. Then the next thing that we're going to talk about is some specific inhibitors that I took the time to just draft in a table. So in complex 1, uh, one of the some of the inhibitors are rotenone, we have amytal and pyridine. In complex 2, you have carboxyl, malonate, as well as oxaloacetate. In complex 3, you have antimyosin, and antimyosin A is actually an antibiotic. So in complex 3, it's inhibited by antibiotics, which is methazole. Okay, so we have antimyosin and methazole, as well as complex 4, which is inhibited by cyanide that you can see over here, as well as hydrogen sulfide, as well as sodium azide. Okay, so these compounds like cyanide, hydrogen sulfide, and carbon monoxide as well. Let me just include that. So, carbon monoxide, what they do is that they block the electron flow through complex 4 at this point. So, here I'm just concentrating on complex 4. So, they block the electron flow through complex 4. And these compounds, they prevent the passage of the electrons by binding to the component of the electron transport chain. Alright, so they prevent the oxidation reduction from actually happening. Now, if oxidation reduction is incomplete, what you're going to have is you have reactive oxygen species, ROS, or a superoxide. So this superoxide is quite dangerous because it can damage DNA as well as proteins. Okay, because it can disfigure their shape, and if the shape is disfigured, even the function is going to be affected. So the reactive oxygen species damage, as it damages the DNA, the proteins, it also causes lipid peroxidation. Alright, so enzymes such as the super oxide dismutase, or the catalase, or the glutathione peroxidase, these are the ones that act against those reactive oxygen species. The next thing that we're going to talk about now is oxidative phosphorylation. How exactly is ATP formed? That's what we are looking at now. So this is oxidative phosphorylation. So under oxidative phosphorylation, we're talking about the generation of ATP. Okay, so there is ADP being added to an inorganic phosphate in order to form ATP. But how exactly does this happen? Alright, so there is what we call a proton pump. So during electron transport, there is a pumping of hydrogen atoms, which are the protons. Remember, this is a proton carrier. There's a pumping of hydrogen protons across the inner mitochondrial space. So if this is the this is our domain, you have many protons that are outside, but now they have to cross over to come inside the inner mitochondrial space. Alright, so as they cross over from the matrix to the intermembrane space at complex 1, 2, complex 1, complex 3, and complex 4. It's coupled with the synthesis of ATP. Okay, like we had been saying, there's a pumping of electrons, movement of protons, and whatnot. So the process is what creates this process of movement of protons from one place to another creates an electron, or rather an electrical gradient, or a pH gradient. Okay, there will be more protons at one end than the other. So the energy that's generated by this proton gradient is the one that gives the whole process sufficient drive for the ATP synthesis. Okay, so now we're going to go into talking about our ATP synthase. Okay, okay. So under the ATP synthase, this is what was drawn. So this is what is referred to as ATP synthase. So the ATP synthase, the enzyme complex of the ATP synthase, which is complex 4, I mean complex 5, is the one that synthesizes the ATP using the energy of the proton gradient that we had talked about, that's generated by the electron transport chain. Okay? So the ATP synthase has two domains. It has the F0 and the F1 domain, which act as the rotational systems, like for instance, like the Kariba Dam, right? There's what we call a motor. So as water is passing, it turns these turbines, okay, and these turbines are what drive and make electricity. So the same thing is happening in this case. All right. So what exactly is happening? So um, the chemoisosmotic hypothesis proposes that the protons actually re-enter the matrix by passing through this channel into the membrane. In the membrane, so they pass through this channel, right? And when they pass through, the F naught rotation causes this F1 domain to change its conformation. All right. So when the conformation is changed, or rather conformation, this is what allows ADP to bind to an inorganic phosphate to give us ATP. So there is one more thing. There's one more inhibitor. We call it oligomycin. Oligomycin binds to the F naught. This is what oligomycin does. It has the ability to bind to the F naught domain. Thereby, if it binds to the F domain, it closes the channel through which these protons are able to enter through the into the mitochondrial matrix. Thereby, this is what causes prevention of the oxidative phosphorylation. And if that's prevented, it means no ATP occurs. All right. All right. So the next thing is that we're just going to talk about is energetics of the aerobic respiration. All right. So the first component we talk about is glucose. So glucose goes into the glycolysis. The end product gives us two pyruvate molecules, and these two pyruvate molecules, there's formation of two NADH molecules. Okay. Now, pyruvate can also it undergoes oxidation reduction reaction to form acetyl CoA, which then enters into the Krebs cycle, or commonly known as the citric acid cycle. Now, in the process, it forms two NADH molecules. All right. So, in the Krebs cycle, or the citric acid cycle, we have two FAD molecules that are formed, as well as three NADH molecules that are formed. And remember that this is occurring twice, that's why it's written 2x, 2x, as well as two GTP molecules that are being formed. All right. Then in stage 3, we are just talking about the number of NADH molecules that are formed due to oxidative phosphorylation, and the total is 10. Okay, 10 NADH and 2 FADH. Okay, now for every NADH molecule that's formed, we have 2.5 ATP molecules that are formed. And for every FADH molecule formed, we have 1.5 ATP molecule that's formed. So we multiply the 10 molecules times the number of ATP. It gives us 25. And there it gives us 3. So in total, under oxidative phosphorylation, what you're going to have is a total of 32 ATP molecules that are formed as a result of oxidative phosphorylation. Okay, so this is what was going on. All right. Okay. The next thing that we'll talk about is the uncoupling proteins. Okay. So the uncoupling proteins, denoted with UCP, these occur in the inner mitochondrial membrane of mammals. So here we're talking about production of energy using a different means. So these carriers, these carrier proteins are referred to as a proton link. Sorry, they're referred to as proton link. Why? Why are they referred to as a proton link? This is because they increase the permeability of the inner membrane to protons. Okay, so they are increasing the permeability of the inner membrane to these protons. However, without energy being captured, so protons are entering the inner mitochondrial membrane, but there is no energy being captured as they pass, which is the ATP. Okay, now that energy is released as heat. Okay, and the process, this process is called non-shivering thermogenesis. So it's called non-shivering thermogenesis. Okay, so this means protons are being transferred via the inner mitochondrial membrane, right? But as they are being transferred, energy is not being captured. Okay, and as they pass, they release heat. So the uncoupling proteins one, it's called thermogenin. Okay, one of these proteins, sorry, is called thermogenin. Okay, so thermogenin is the one responsible for the heat production in brown adipocytes. Okay, of animals or of mammals. So um, brown fat uses almost about 90% of its respiratory energy for this thermogenesis. Okay, this is to tell you that most of the brown fat is used in the production of energy. All right, and in response to the code in the neonate, which is children, as well as those animals that hibernate during the winter. So in human beings, to have little brown fat, except in the newborn, sorry, and the uncoupling protein one, it doesn't appear to play a major role in the energy balance. Okay, because when you look at this brown fat, it doesn't play as that much of a major role in providing energy. I mean, and in maintaining the balance of energy. All right. So when we have this synthetic meaning those that are not formed by the body. For instance, we have 2,4 dinitrophenol. This one is synthetic. It's as though it's made in the labs. Or dicumaro. Okay, so the several that added to cells to stop it stops ATP This one, it stops ATP synthesis. Okay, sorry. So it stops ATP synthesis, but the electron transport chain continues. The ETC continues. All right. So the DNP, which is this one, I'll just denote it as DNP. The DNP and the other uncoupling agents are actually lipid soluble. All right. The lipid soluble molecules that can bind hydrogen ions and transport them across the membrane in what we call ionophores. Okay, this is what's happening. Right, so these ionophores that are formed, uh, they prevent formation of the proton gradient. All right, so they prevent formation of the proton gradient since no proton gradient forms. Okay, there's no formation of the proton gradient. It means there'll be no ATP generated by the oxidative phosphorylation reaction. So in high doses, aspirin and silicates, okay, aspirin and salicylates and couple oxidative phosphorylation. Okay, so this is the end of the lecture. Hope you enjoyed it.